and welcome back. So this is Shruthi and today in this video we'll be dealing with a chapter of zoology which is excretory products and the elimination. So coming to the excretory products and their elimination. Now a human body or any kind of animal body, what it does is it will accumulate certain kind of products such as the ammonia, urea and the uric acid. So along with these nitrogenous weights, there is sodium, there is potassium, there is chlorine, there is phosphate. Now these are the various kinds of materials which are getting accumulated in your body. They are either formed by the ingestion or by the metabolic activities. Now in order to perform the functions, a proper function of your body, so these products whatever are there, these are the major nitrogenous waste, ammonia, urea and uric acid, has to be removed partially or fully. They have to be removed partially or fully for the proper functioning of your body. So now we'll discuss about how ammonia, urea and uric acid and these materials are excreted out of your body. Now coming to the very first, ammonia. Now there are organisms or the animals which secretes ammonia. Okay. So they have major amount, these are the substances which are the major nitrogenous waste, okay. Ammonia, urea and the uric acids are considered to be major nitrogenous waste. So in this chapter we will be dealing how these actually ammonia, urea and uric acid are actually eliminated from our body. So coming to the ammonia. Now ammonia is secreted by, excreted by certain animals such as aquatic insects, aquatic amphibians and the bony fishes. Now these animals, okay, aquatic insects, aquatic amphibians and the bony fishes will excrete ammonia, okay. So they will excrete ammonia through the gills. They are aquatic, right? So they will have, they'll secrete the ammonium cells, hence they are referred to as ammonialism, and the organisms which are performing this are referred to as ammonialism, okay. So this is about the ammonia. Now coming to the urea. There is some terrestrial adaptation. They have got few modifications. So few terrestrial animals and marine fishes. Okay. Few terrestrial animals and marine fishes, what they will do is they will excrete out the urea. Now the ammonia which is there in their body. Okay. The ammonia which is there in their body is getting converted to urea. Okay. So these occurs in the liver. So they'll have a liver, from these ammonia is getting converted to urea and some terrestrial animals and the marine fishes. So these occurs and they will excrete out the urea. So this will maintain the osmotic regulation and helps in maintenance of ionic balance. Okay. So this is the thing which will happen in the some terrestrial animals and certain marine fishes. Okay. So this is with the bony fishes. This is with the marine fishes. Now coming to the uric acid. So there are reptiles. Okay. There are reptiles and birds. So they will excrete the uric acid. Now how this is done is they will secrete this uric acid. Then the uric acid which is secreted by them will also help in the maintenance of ionic balance. Okay. So these are various kinds of animals which will release ammonia, urea and uric acid. If the animals are releasing ammonia, they are referred to as ammonotolic. If they are releasing urea, so they are ureotolic, and uric acid are referred to as uricotolic. This is some terrestrial adaptations. These are done by reptiles and birds, and ammonia excretion you can see in some aquatic amphibians and aquatic insects. Now coming to the excretory organs of various, like if you talk about the invertebrates, they have really simple excretory kind of system. And when you talk about the vertebrates, they have a complex kind of excretory system which is like tubular in structure. So there are photonephridia or called as the flame cells. Okay. So photonephridia or the flame cells are the cells which are present in the platyhelminthes such as the rotifera. One example is rotifera. So you can see these kind of excretion in platyhelminthes, annelids and cephalopods. Okay. So this is about the photonephridia. Then there are cells called as nephridia. So these nephridia kind of excretory organs you can see in earthworms, and you can also see in few of the annelids. Now coming to the Malpighian tubule. Malpighian tubules are the excretory organs which are present in insect, including the cockroach. So these kind of excretory system you see in most of the insect, including the cockroach. Now green glands or the antennal glands. These are the excretory organs which are present in crustaceans. Crustaceans such as prawns. In prawns you can see the antennal glands or the green glands. So these are the various kinds of excretory organs which are present in various kinds of invertebrates. Now considering to the human being. Human being have a very complex excretory kind of system. So we'll be dealing with that. Now human excretory system, it consists of a pair of kidneys. Okay. Then a pair of ureter and the bladder. So these are the parts of the human excretory system. Now coming to the dimension of kidney, if you talk a normal dimension, an average healthy human being, the length of the kidney will be 10 to 12 centimeters and the width will be 5 to 7. The thickness will be 2 centimeters. On an average, weight will be 120 to 170 grams. Okay, so this is the dimension of an average human kidney. Now after this kidneys, the human kidney which is there, it has got two parts, the outer cortex and the inner capsule. The outer cortex which is there, it is covered in a tough capsule-like structure which is called as the renal capsule. Okay. So as I have told you, outer cortex and the inner medulla. Now coming to the outer cortex, this cortex is present in between the medulla. So this is the medulla region, right? So the cortex is present in between the medullary pyramids. So these are the medullary pyramids. So this is whatever the part is there, this is called as the cortex. This will form the medulla region. So renal cortex, this cortex region is present in between the medullary pyramids, which is called as the renal column. So these renal column will form again column of Bertini. Okay. So you have got with the outer cortex which is surrounded by a tough capsule-like structure which is called as the renal capsule, and inner structure you have a medulla. So these medulla is again, it consists of conical projections like called as the renal medullary pyramid. Now these medullary pyramids have a projection over here which are referred to as calyx. Okay. So this is the outer covering of the kidney which is called as renal capsule. This will be the cortex, this will be the medulla, this will be the conical projection called as the pyramid, medullary pyramid. Between these medullary pyramid you will find the outer cortex region, and these are the calyx which are the projections. Now at the concave surface, inner part of the center of the cortex of the kidney, there is a structure called as the hilum. Now this hilum is a part where the renal artery, where the renal vein and also certain blood vessels and the ureter will enter. Okay. So hilum is a region where the renal artery, renal vein and certain other blood vessels will enter and the ureter will come out. And this is thrown out into a conical broad funnel-like structure which is referred to as renal pelvis. Okay. So these renal pelvis has a projection called as the calyx. So this is about the basic structure of the kidney. So now the kidney which is there over here, we have completed with the kidney. It also consists of a structural or the functional unit called as a nephron. There are about 1 million of nephrons in each of the kidneys. Okay. So now these nephrons are the structural and functional unit of the excretory system. So how these nephrons are formed? Again, this nephron is divided into glomerulus and the renal tubule. So nephron has the two parts, that is glomerulus and the renal tubule. Now these glomerulus, it is a tuft of capillaries, means a bunch of capillaries. These capillaries are coming from the renal artery. So renal artery will be there. You have renal artery means again you have afferent and efferent, right? So it is a tuft of capillaries of these afferent arterioles. And the blood will be taken away from the glomerulus by these efferent arterioles. Okay. So this is about the glomerulus part. It will also help in the maintenance of ionic balance and the osmoregulation. So this is about the glomerulus part. Now coming to the renal tubule. Now renal tubule, it consists of Malpighian body. This Malpighian body, or we call it as the renal corpuscle. So now this renal tubule is Malpighian tubule. So this renal tubule, it has a double-walled cup-like structure, which is called as Bowman's capsule. Okay, it has a double-walled cup-like structure called as the Bowman's capsule. Now this Bowman capsule will surround this glomerulus. So glomerulus along with the Bowman's capsule is referred to as Malpighian body. So Malpighian body, once the Malpighian body is done, this Malpighian body will lead to a highly coiled structure tubule called as the PCT. Then these PCT will lead to a hairpin-like structure called as the loop of Henle. Okay. So Malpighian body is formed by this glomerulus plus Bowman's capsule. So that will form the Malpighian body. Malpighian body will finally lead to a highly coiled tubular-like structure, that is the PCT. So once the PCT is formed, so these PCT is highly coiled, right? Then that will lead to hairpin-like structure called as the loop of Henle. So loop of Henle will have two limbs. One is ascending and the other is descending. So these ascending loop of Henle will again lead to highly coiled structure, which is called as distal convoluted tubule, DCT. So these DCT will again finally lead to a straight tubular-like structure called as collecting duct. So once it reaches the collecting duct, after the collecting duct, this will lead to a renal pelvis. Renal pelvis, again the outer cortex, inner medulla region, finally leading to conical projections such as the calyces. From there, it will pass out through the ureter. So this is how actually the procedure occurs in the kidney, right? So Malpighian body is leading to PCT. After the PCT, it is getting to loop of Henle. Then you have two limbs, ascending and descending limb. So descending will be here. Ascending will lead to DCT. Then that will lead to collecting duct. Then it will reach to renal pelvis, outer cortex region, medulla region, calyces, then ureter region. Okay. So this is the basic structure. Now what is that? So PCT plus DCT and glomerulus part. These are the regions of cortex. So these you will find in the cortex region. Whereas the loop of Henle is the structure which is present in the medulla region. Okay. So now most of the human kidneys, they have very short loop of Henle. If when they have a shorter loop of Henle, so they will extend only up to the cortical region. Okay. They'll go a very little into the medulla region. So most of the loop of Henle is present in the cortical region. So where in such cases, those are called as corticular nephrons or the cortical nephrons. Because they extend, most of the part is extending only into the cortex region. Whereas coming to the few persons, they have a really longer loop of Henle, so that will extend into the medullary region. When that loop of Henle, which is longer, extending into the medullary region, such loop of Henle are referred to as juxtamedullary nephrons. Okay, so those are refers to as juxtamedullary nephrons. So this is about the basic structure of the kidney. Now these afferent arterioles, which are afferent and efferent arterioles, which are carrying away the blood away from the glomerulus. Okay, so which are carrying away from the glomerulus, they will form a tubular-like structure. So those tubular-like structure is referred to as peritubular network of capillaries. Okay, so that is referred to as peritubular network of capillaries. Now these peritubular network of capillaries will reach again, this will run in parallel to the descending loop of this Henle. So descending loop of Henle, it will run parallelly and form a U-shaped structure called as the vasa recta. So when coming to the structure, so here you will have a kidney, right? So this will be the glomerulus part. Here you will get the afferent arteriole. Then this will continue down. So here you will get PCT. Then This PCT will lead to loop of Henle over here. Ascending loop will go up here, DCT, then this will lead to a straight collecting duct. Okay, so this is the structure, right? So now these afferent arteriole will come from over here and run parallel. It will run parallel to this and form a U-shaped structure. So this will be U-shaped structure like this. So this part is called as vasa recta. Okay. So this is a structure which will run in parallel to this peritubular network of capillaries. So with this is a basic human excretory structure and how the actually one structure will lead to the another. So in the next video we'll be discussing about the mechanism of urine formation, how does it occurs, and after that we'll be dealing with the function of the tubules as well. Dealing with the micturition. So first we'll deal with the urine formation. Now coming to the urine formation, there are about 3 steps involved in the urine formation. The very first step is glomerular filtration. Then the second step we have is the reabsorption, and the third step we have is the secretion. Okay. So these are the 3 steps which are involved in the formation of the urine. Now first we will be dealing with the glomerular filtration. Now glomerular filtration, that is about 1100 to 1200 of ml, 1200 of urine is produced. Okay. So now the 1100 to 1200 ml of urine is produced, right? So now this is about 1/5 of the blood. Okay. This is about 1/5 of the blood which is pumped out by the ventricular musculature per minute. Okay. So this is the thing about the glomerular filtration. It is a very first step in the filtration. Okay. So the filtration of the blood is a very first process which is performed by the glomerulus. It filtrates about 1100 to 1200 ml, which is about 1/5 of the blood which is pumped out by the ventricular musculature. Now these filtration, it occurs by 3 different layers. So these 3 different layers are the endothelium of the glomerulus. Okay. As the glomerulus is performing the function, the endothelium of the glomerulus is the function. Then epithelium of the Bowman's capsule and the basement membrane in between them. So these are the 3 different layers which will perform the function of the glomerular filtration. Okay. Endothelium of glomerulus, epithelium of the Bowman's capsule and the basement membrane in between them. Now these epithelium of the Bowman's capsule has specialized cells called as the podocytes. Okay. They contain the specialized cells such as the podocytes, and these cells are arranged in such an intricate manner so that they have space in between them. The space in between them is called as slits. Now these slits, what they will allow is, they will allow almost the passage of all the nutrients or the elements which are present in the plasma. So they will allow all the essential elements to pass through them, leaving the larger proteins behind. Only except the plasma proteins, all the elements are being passed through this podocyte. Hence, this process is referred to as ultrafiltration. Okay. So they are arranged in such a manner because of these elements, entire blood is getting purified. Only the larger proteins are left. So hence this process is referred to as ultrafiltration process. Now coming to the glomerular filtration rate. It is the amount of filtrate formed per minute. Okay. So this is the amount of filtrate formed per minute. So this is about 180 liters of filtrate is formed per day. Now the amount of filtration which is formed. So to maintain this, there is a inbuilt mechanism in our body. So there is an inbuilt mechanism in our body to maintain this filtration rate or to maintain the regulation of glomerulus. So this mechanism is referred to as juxtaglomerular apparatus. Okay. So juxtaglomerular apparatus. This is a special apparatus which is inbuilt in our body for the regulation of the glomerular filtration. Now these juxtaglomerular apparatus, which is the modification of distal convoluted tubule and the afferent arteriole. Afferent is the sensorial arteriole, which is again a part of the renal artery. Okay. So due to the modification of these, that is the juxtaglomerular apparatus. Now how these apparatus is getting stimulated, right? Regulation means it has to get stimulated, right? So the decreased levels of glomerular filtration rate will stimulate the renin, and once the renin gets stimulated, that will cause the GFR get back to the normal conditions. Okay. So this is the thing about the glomerular filtration. Now next we'll be dealing about the reabsorption process. Now reabsorption process is done by the tubular system. Okay, the entire tubular system of the kidney, that is PCT, DCT, loop of Henle, convoluted tubule, that will perform the function of reabsorption. So when you compare the amount of filtrate, okay, and the amount of filtrate formed is 180 liters per day, and when you talk about the excretion, only 1.5 liters of urine or the filtrate is excreted out. So that means about 99% of the tubular reabsorption take place in our body. It has to take place. So 99% of the tubular excretion or tubular reabsorption is done. So 180 liters is produced, 1.5 liters is excreted out. So this is done by the tubular part of the excretory system. So now these tubular reabsorption, the tubular part which is getting the reabsorption in different segments of the nephron, it occurs by different methods. Somewhere it occurs by active means and somewhere it occurs by passive means. Now what are the things which are getting actively reabsorbed, such as glucose, sodium or the amino acids, which are a little complex materials. So these needs a little energy with the help of a carrier protein or something. So with the help of the active energy, they are getting transported. Whereas nitrogenous waste, they don't require more of the energy. So they are easily, passively, they are getting transported. Now these reabsorption, as I have told you, tubular epithelial cells will perform the function of reabsorption, and they are present in different segments of nephron. Different segments of nephron, they will perform different way of transportation. So these also in the reabsorption, here you have done with the glucose, amino acids and the nitrogenous waste. The reabsorption of water also takes place in the initial level of the initial segments of the nephron. Passively it takes. And there is also a secretion of hydrogen ions, potassium ions and the ammonia ions. Okay. So these secretion plays a very important role. Because of the secretion of these hydrogen, potassium and ammonia, the ionic balance of the excretory system is maintained, and it is also helping in the maintenance of acid-base balance of the fluid. Okay. So this is the thing about the reabsorption. As I have told you that the reabsorption occurs by the tubular part. So tubular part consists of PCT, then you have a hairpin-like structure called as loop of Henle, then you have DCT, then you have the collecting duct. So first we will talk about the PCT. Now these proximal convoluted tubule, it is lined by simple cuboidal epithelium cells. Now as these are cuboidal cells, this will increase the surface area of the cells. Okay. So when the surface area increases, there is more of the surface area, means more of the reabsorption occurs. So about 70 to 80% of the electrolytes or the H2O, whatever is there, is getting absorbed in the PCT. So it is a major site of reabsorption. That means 70 to 80% of the electrolytes and water is getting reabsorbed over here. So this will help in the maintenance of pH, also the ionic balance of the filtrate and the body fluids. So at the same time with this, the absorption of HCO3 also occurs, the secretion of hydrogen ion, potassium ion and the ammonia. So these are secreted and the absorption of HCO3 occurs. So PCT is a major site of reabsorption, and 70 to 80% of the reabsorption occurs. This is because only the simple cuboidal epithelial cells, they will increase the surface area for reabsorption. Next, proceeding to loop of Henle. This is a hairpin-like structure, right? So this will loop of Henle, what it will do is, the structure of loop of Henle is not really meant for reabsorption. Very little reabsorption takes place here, but this will increase high osmolarity. Okay, so here you will have a really higher osmolarity. Now as I have told you, loop of Henle will have ascending limb and the descending limb, right? So ascending limb will go upward, descending limb will come downwards. So this ascending limb, when I talk about this is permeable to H2O. That means it will allow the passage of H2O. Okay. So this is impermeable. So this ascending limb, it will not allow H2O to pass through it. But however, it will allow the passage of certain electrolytes either actively or passively. Okay. Now coming to the descending limb. This is permeable to H2O. Okay. Permeable to H2O means this will allow the passage of water, and impermeable to electrolytes. So this will not allow electrolytes such as sodium, potassium to pass through that. So there is a very little reabsorption taking place here. Now this here, it is permeable to H2O, right? Descending loop of Henle is permeable to only H2O and impermeable to electrolyte. So the filtrate will move downwards. As it is impermeable to electrolytes, so the filtrate which is there, it will move downwards. And here, which is impermeable to water, ascending limb, the filtrate move upwards. So due to this, the concentration gradient is maintained. Once the concentration gradient is maintained, due to the up and down of this, the fluid get diluted, because once the passage of electrolytes moves to the medullary fluid, right? The fluid is getting downwards and it will move to the medullary fluid. The fluid will get diluted. Like this, in this way, the concentration gradient is maintained in the loop of Henle. Next, we will be dealing with the DCT. Now, DCT is maintained for conditional reabsorption. When if there is a condition where the PCT can't perform the function of loop of Henle, then there is DCT to perform this function. So here reabsorption of sodium ions and the H2O occurs, and there is also, it is capable of absorption of HCO3. Also there is a secretion of hydrogen, potassium and the ammonia. By the secretion of these H2O, H plus and K plus and ammonia, the pH and the sodium potassium balance is maintained in our body. Next we will be dealing with the collecting duct. Now this is a straight tube-like structure, okay, like this. It will extend from the cortex region to the medulla region. Now this is actually meant for the water reabsorption. So how the water is reabsorbed is the increase. So water reabsorption occurs here more and more to produce the concentrated urine. Most of the water get reabsorbed, so the concentrated urine is produced. And these will, what it will do is, it will allow small amount of urea to pass through the medullary interstitial fluid, and this will maintain the osmolarity also. So by the secretion of this H plus and the potassium, pH and the ionic balance is maintained in the collecting duct as well. So most of the thing here important over here is PCT. It is 70 to 80% of absorption, and loop of Henle, you should remember the fluid is getting diluted because movement of upwards and downwards, concentration gradient is maintained. DCT mein you have conditional reabsorption, ionic balance is maintained by secretion of H plus and potassium and ammonia. Same occurs with the collecting duct, okay, but here concentrated amount of urine is produced. So this is about the urine formation and the function of the tubules. Now let us discuss with the mechanism of concentration of the filtrate. So mechanism of concentration of filtrate. So humans have the ability, they have the ability to produce the concentrated amount of urine. So they produces the concentrated filtrate. Now the concentrated amount of filtrate or the concentrated filtrate, to produce this concentrated filtrate, loop of Henle and vasa recta plays an important role. So the loop of Henle has two limbs, right, ascending and the descending. So during in ascending loop of Henle and descending loop of Henle, the flow of filtrate is in reverse direction, that is in the opposite direction. One moves up in the descending loop, it moves down, vice versa. So due to that, it helps in maintaining the counter mechanism. Right? So loop of Henle, two limbs, the flow of filtrate is in reverse direction, that will cause the help in the current counter mechanism. The same thing happens with the vasa recta. It also has two limbs and the flow of filtrate is again in the opposite direction, that will again help in the maintenance of counter current mechanism. So the proximity between loop of Henle and vasa recta along with this counter current mechanism, this will help in the maintenance of osmolarity in the inner medullary, okay, inner medullary part of the kidney. So once this is done, this proximity, what it will cause is osmolarity is maintained from 300 nanometers of cortex. In the cortex region, it is 300 nanometers, and whereas this will go and increase to 1200 nanometers in medullary region. So coming to the this osmolarity, the gradient is maintained by the sodium chloride and the urea. Now the sodium chloride, it is getting transported to ascending loop of Henle of, okay, ascending loop of Henle, okay, loop of Henle has two things, ascending limb and the descending limb. So that sodium chloride will come here to ascending limb. From the ascending limb, it will reach the descending limb of vasa recta. Okay, so from the loop of Henle to the vasa recta descending limb. Then once it reaches the descending limb of the vasa recta, again it has to return back, right? So to return back to the interstitial fluid, this gets by the ascending limb of the vasa recta. So after the descending limb of the vasa recta, it reaches the ascending limb of vasa recta to get back into the interstitium. Okay, so this is about the sodium chloride. Now same thing happens with the urea. First it reaches the ascending loop of Henle. From the ascending loop of Henle, this has to get transported to medullary part. So how this reaches the medullary intestinum part is by the collecting tubule. From the collecting tubule, with the help of this collecting tubule, it will reach the medullary interstitial part. Now, due to this, the special system, whatever is maintained, the transport mechanism is formed by a special arrangement. So, these special arrangement consists of these loop of Henle, vasa recta, and the counter current mechanism. That is the flow of the filtrate in opposite direction. So, that will form a special structure. So, these will maintain the mechanism of concentration of filtrate. So this concentration is maintained. The process, whatever is there, this special arrangement will maintain the concentration gradient in the medullary interstitium. That is the medullary region of the kidney. So these mechanism or the concentration gradient which is maintained in the medullary part, this will allow the easy passage of the water from the collecting tubule. From the collecting tubule, the water is easily passed because of this concentration gradient. And at the end, due to the easy passage or the easy flow due to the counter current mechanism, it will lead to the production of concentrated fluid. Okay. So humans have the tendency to produce the concentrated fluid, right? So in this way, due to the passage from ascending loop of Henle to the descending loop of vasa recta, and concentration of the fluid is produced and maintained. So now human at the initial level, whatever the fluid is formed, they have the tendency to produce the fluid more concentrated. So they can produce 4 times of the concentrated fluid at the, whatever the initially fluid is formed, they have the tendency to produce the fluid 4 times more concentrated than the initial level. Okay. So this is the mechanism how the concentration of the filtrate is maintained. The counter current mechanism is a major thing which occurs over here, which is maintained by loop of Henle and the vasa recta. So next we are going to deal with the micturition. Now the urine is formed. Okay. So urine is formed in the nephron. Reabsorption is done. Secretion is done. Now the entire urine is formed in the nephron. So once the urine is formed, it reaches the bladder part. The bladder part will receive the whatever the filtrate or the urine is formed, and it will get stored there. Okay. So bladder will store the urine till it gets the voluntary signal from the central nervous system. Now once it gets the voluntary signal from the central nervous system, that will cause the stretch of the bladder. Then the stretch of the bladder, what it causes is, the urinary bladder, it is already filled with the urine. So once it is filled with the urine, that will cause the stretch of the bladder. So after that, the bladder has a certain receptors on its, this will cause the stimulation of central nervous system. Now these central nervous system, once it receives the signals, it will cause the motor signaling. Once the motor message this gets initiated, that will leads to the contraction of the muscle of the uterus. So here the contraction occurs, then the urethral muscle, whatever is there, that will start getting contracted. Simultaneously, the relaxation of the sphincter occurs. So now the contraction of urethral muscle occurs, then these will lead to release of the urine. So this entire process is referred to as micturition, and the entire neural mechanism is referred to as micturition reflex. Now the urine which is formed is slightly yellow, watery color fluid which is acidic and has a characteristic odor. Now the amount of urine that is formed in our body is 1 to 1.5 liter per day. Okay. So the amount of urine which is excreted out per day is 25 to 30 grams, and the certain conditions which will affect the excretory system can cause alterations in the kidney. Okay. So these are the various conditions causing the production of kidney and leading to alteration. Now the diagnosis, how it occurs is, the diagnosis or the certain changes in the condition can help you in the diagnosis or the knowing of certain malfunctioning of the kidneys. Okay, so abnormal conditions will help you know in the malfunctioning of the kidneys. So with this we have completed the urine formation, function of tubules, mechanism of concentration of filtrate and the micturition.